Hello, everyone, and welcome to a very special end of season International Online Spinal Cord Injury Research Seminar. My name is Bethany Condilis, and with me is Matthew. Actually, Matthew, I don't know how to pronounce your last name.
It's Rodreick.
It is Rodreick, like it's spelled. Okay, Matthew Rodreick. Yeah. Unite to Fight Paralysis. We are very excited for this event today. Thank you so much for joining us. We have an amazing lineup of speakers, and we're so happy to be working with Unite to Fight Paralysis on this. So we're going to, So I'll give Matthew a second to kind of go for a bit of an overview and intro for the day before he introduces our first speaker. We do have a live chat, which we hope everyone will make use of, and we're looking forward to a really great session today. Okay, it's all you, Matthew.
Thanks, Bethany. So again, I'm Matthew Rodreick. I'm the Executive Director at Unite to Fight Paralysis. We're a grassroots SCI-focused organization that is explicitly focused on expediting curative interventions for spinal cord injury. And so today, obviously, we have a few guests that I hope you know about already. And I'll give you a little background on us and particularly our relationship with IOSCIRS. Both of us really focused on helping to engage increasingly folks with SCI, people who live with the injury, and researchers, those who research the injury. And also just a quick plug for our annual Science and Advocacy Symposium, which is coming up October 22nd and 23rd. Bethany and folks from IOSCIRS will be presenting, along with a whole host of scientists and clinicians really focused on the cure space and research space. So everyone from Gregor Cortine and onward to Dr. Michael Lane at Drexel College of Medicine, a handful of strategies focused on restoring function for spinal cord injury. It'll be in Salt Lake City. You can go to u2fp.org and find out as much information as you like. But it's very similar to what IOSCIRS is trying to do with seminars like this, which is increase the dialogue and engagement between people who live with the injury and those who research it. And so we're really grateful to be here as part of that effort today, both today and then also having them at our symposium. And for those of you in the community who don't know, this International Online Spinal Cord Injury Research Seminar has been going on for quite a while. It was really a response to COVID and the lack of opportunity for researchers to gather together at conferences where they do meet each other and share and develop collaborations. And it's been a really successful way, in the absence of some of those meetings due to the pandemic, to be able to get together and confer and learn from one another and develop potential collaborations. And then lastly, the goals for this session are really focused on delivering these three scientific presentations from Dr. Hill, Dr. Magnuson, Dr. Moritz in a more lay-friendly format, and then really encouraging you. So really pay attention. We really want to get questions from you. We really want to have a conversation post their presentation that involves you from the spinal cord injury community. Bethany? Also quickly, I'll just give you a quick rundown. I forgot I've got more slides on U2FP. So this is just a little bit of what we do. I already mentioned our symposium coming up this fall. We also have a scientific advisory board that we offer as a service to usually small, mid-sized spinal cord injury foundations that really lack the expertise to vet funding proposals. We also have a couple of marathon teams in Chicago and the Twin Cities, here in Minnesota, and also a virtual run. It really helps us to raise awareness about spinal cord injury, but also raise funds for our work. And then lastly, something you might want to check out most recently, we have a regular podcast, and most recently we interviewed Bethany Condilis and Megan Detloff, who are responsible for IOSCIRS, part of the team. We interviewed them about what they're doing, but we've interviewed lots of different scientists and advocates throughout the space. And then lastly, we have what we call our Cure Advocacy Network, where we have passed bills in multiple states to designate and target funding for spinal cord injury. And now it says 12 million, but we're actually now close to 20 million cumulatively between Ohio, Pennsylvania, Minnesota, also Washington, and we currently have an effort in Wisconsin. So if anybody out there lives in Wisconsin and doesn't know about it, we'd love to hear from you and love you to join us. And you can find out more at u2fp.org.
Okay, thank you, Matthew. And just a reminder, all of the links that we're talking about and we will be talking about throughout the session are in the description, and we'll be dropping them in the chat for you as well. So as Matthew mentioned, we are the International Online Spinal Cord Injury Research Seminars, which, as he said, was born out of the pandemic as a way to stay in touch and to continue to give high quality technical science talks to other scientists. But one thing we've realized is we have this very unique platform. We are online and we are totally free. You don't have to pay to go to a conference. You don't have to travel. And we think an important part of what we do is learning to engage with non-scientists as well, who are also invested in the work we do to repair and improve recoveries from spinal cord injury. To that end, we've started doing some things with our normal programming. At the end of every talk, we ask our speakers to put up a layperson summary, so that if you want to come and learn about cutting edge science, but you're going to get bogged down maybe in some of the technical details, you can get a big take home message. So we do try and make sure that every seminar has something that everyone can take away from it, even if you're not going to be, really deep and gritty in the methodology or whatnot. And then also we try and engage on social media. So we have efforts to post research synopses and keep people posted on ongoing work on our Twitter. So if you're not following us on there and you want to check that out, we welcome you to do that. So here we're going to hand it back to Matthew for engagement and communication.
So again, as I mentioned, one of the critical pieces for today, especially, and definitely where IOSCIRS would like to continue and where we've been operating for a while, is really trying to develop effective relationships between researchers and people who live with the injury. And in addition, for those of you who do not know about the North American SCI Consortium, also you should look for that link. That's formed just a few years ago. I actually was one of the executive council members for a couple of years, along with a number of colleagues that you may be familiar with, Dr. Kim Anderson and John Chernesky from Praxis and Sasha, others. I won't list all the names, but NASCIC is very, very focused on providing access to the community, both to the research community, the clinical community, even industry, to help develop our voice. And again, I mentioned our annual symposium and our Cure Advocacy Network. Those two are very much driven by the same thing. So I earlier mentioned money in state funding proposals, that we're close to 20 million. That's really not the key. The key is that we place people on advisory boards to help prioritize and promote discussions between researchers, clinicians, and people who live with the injury, so that everybody understands one another. People with the injury understand the science better. Scientists understand what it's like to deal with the injury, and then use that dialogue to prioritize and fund science. And so that's very much a part of what we'd like you to check out those links, get involved with us, get involved with NASCIC. There are plenty of opportunities to do that. Would you like me to keep going, Bethany?
You're doing such a great job. So go ahead, or I can take over. It doesn't matter.
Go ahead. No, go ahead.
So yeah, today is going to be really fun and dynamic. The goal is to give people a glimpse into some cutting edge science from some really great researchers who are here today. So we will kind of give you some hints into things, active work in their labs. And then also just some commentary about spinal cord injury research in general, themes and where it is, where we need to be going, etc. And so, yeah, the goal for today is to be a start of a conversation. We don't want it to be a one-time standalone event. We want this to be ongoing effort and dialogue between researchers and people living with SCI, and to have the IOSCIRS to contribute to the work that U2FP is doing and so many other amazing organizations. So yeah, we want to start this dialogue off today. So please engage in the chat, ask your questions. Matthew will be moderating Q&A sessions. And we also have a survey that we would love for you to fill out at the end to help us figure out how we can do more events like this that'll be useful to other people in the community. So, that's for the end. So we'll take these slides out. And now I'm going to say thank you very much to everybody, remove myself from the broadcast, and bring in our first speaker. Thank you.
Welcome, Dr. Hill. So let me just give you, Dr. Hill, a brief introduction. Dr. Hill comes to us from the Neural Stem Cell Institute since 2019 and has been around the world quite a bit from before landing there all the way back to Toronto, where she gained her master's degree at Queen's University, and then did some training at Dr. Reisman's lab in the UK. The Miami Project to Cure Paralysis, also a visiting scientist at Christopher Dana Reeve, got her PhD at OSU, also at Duke University, and then ended up at the Burke Neurological Institute at Cornell. And now Dr. Hill is here from the Neural Stem Cell Institute. Her lab is focused on regeneration, recovery, restoration of function, but primarily from that, really looking to drive those discoveries towards interventions that will have a meaningful impact for people with spinal cord injury. Go ahead, Dr. Hill.
Great. Well, thank you. Let me just get my slides up. It's always interesting to give a talk when all you're doing is looking at your own screen and you don't have any feedback. But I'm really excited to give this presentation today and to talk about targeting and testing therapies for spinal cord injury repair at the level for non-scientists. So I come from a family where I'm the only scientist, so I'm a little bit familiar with trying to describe what happens after injury to lay people, but it is still always a challenge. So I thought the approach I would take is kind of to introduce the injury and the cells that play a part, some of the work that we are doing in the lab, as well as to highlight other research going on in the field and how we've built upon each other's research and where we are going. And so let me first see if we can get the laser up. There we go. So spinal cord injury occurs when there's damage to the spinal cord, and this is usually a result of vertebral displacement into the spinal canal. It initiates a cascade of events that are highlighted here, involving many different cells and processes, and I'll talk about some of those later on. Ultimately, what happens, though, is you lose motor function, which is the primary outcome that people consider after spinal cord injury. But it also results in a host of other changes to systems in the body. So you lose the ability to move, but you also have altered sensation, you have altered respiration, cardiac events, and bowel, bladder, and sexual dysfunction. And so how does this happen? Well, part of the reason that you get this complex array of problems is because the nervous system is highly organized. You have neurons in the brain that are important for initiating locomotion that send axons down through the spinal cord in this region here, which is called the white matter. It then connects to other neurons in this other part of the spinal cord called the gray matter. And those neurons connect either with more neurons or to their output organs. And so you have this complex array of neurons starting in the brain and extending down through the spinal cord. In many cases, you can think of this a little bit like a power grid. You have an area where signals are generated, you have transmission across power lines, and then you have distribution to houses and buildings and other such things. And so if we look at the spinal cord more closely, what you can see is it's located within the vertebral column. You have axons that exit the spinal cord that are important for those movements and control of the organs. And you have other axons that enter the spinal cord and convey information from your skin, such as pain, sensation, touch. And so if we look even more closely at this region here, what you'll see, if we take a little bit of artistic liberty, is that there's a complex array of cells located within the spinal cord. They're heterogeneous, they have many cellular processes, and they're highly interconnected. And so when the spinal cord is damaged, these cells are impacted. And so we like to think of the spinal cord, as involving these arrays of neurons. And you have the cell body, you have the axon terminals, and you have insulation that's provided. And this insulation is provided by the other type of cell within the spinal cord, the glial cells, in particular this cell type here called the oligodendrocyte. So you have oligodendrocytes and astrocytes, which make up two components of the glia. Astrocytes you can kind of consider these multi-purpose cells. They collect the garbage, they regulate what happens within the system. And then you have another type of glial cell called the microglia, which are the resident immune cells that respond to when there are inflammatory events. There's also the vasculature and the cells associated with the vasculature. So you have this complex network of cells that are present within the spinal cord. And so on a global perspective, we talk about neurons, we talk about glia, we talk about vasculature. But what you can see here from this recent study by Wuhan in Jenny Zhu's lab is that they use this new technology called single cell analysis, which allows us to profile individual cells and mathematically map them into clusters. And what you can see is, yes, there are neurons, there are oligodendrocytes, there are astrocytes, there are myeloid cells, which are their microglia, but there are also many other cells present within the spinal cord. And so this new technology is allowing us to start to understand more the complexity of cells that are present within the cord. Before, we were limited to kind of knowing which markers were present and using histology to look at these cells. Now we can use this new technology to identify these clusters of cells and see that it's much more complex and diverse than previously considered. And it's not just these individual glial cell types that differ, but even if you look within a single population. So if you look at with the neurons within the spinal cord, what you see is that you can cluster neurons. So there's not just one type of neuron within the spinal cord. There are many, many different types of neurons. So you can identify with this technology neurons that suppress activity, that are the inhibitory neurons, neurons that promote activity or the excitatory neurons, as well as the position of neurons within the spinal cord, those that are located at the top, those that are located at the bottom. So this complex array of neurons are even located within the spinal cord. And of course, this schematic shows that there's these differences, but when you look at the primary data from the paper by Samamurthy et al. From Avril Levine's lab, what you see is that it's very, very complex. So within the spinal cord, we think of neurons and glia, but actually there's this complex array of cells. And understanding this complex array of cells that we're only beginning to be able to do is really going to propel the field forward. So as I said, we can think of the nervous system a little bit like an electrical grid, and an injury is much like what happened in 2003 when there was a blackout in the Northeast. A single telephone pole that fell and broke the wire resulted in a blackout of the entire region. And so we all know that when there's a blackout, eventually the power is restored. So how does that happen? Well, we're able to restore power after a blackout because we know the components of the system, or at least the power company knows the components of the system. I've only learned about the components of the system recently. But you have a network that's very clearly outlined. And so when there's a power failure, the company then knows what to do. They check the generating facilities to bring the power back online. They repair the transmission lines. And then they connect the individual service lines. So they're able to repair the system in a highly organized fashion. Although we all know that when the power goes out, it never seems to come back on as quickly as anyone would like. But eventually it comes back on. And so this is able to happen because we know what components are broken and we know what needs to be replaced. But in the spinal cord, we have these large knowledge gaps. So there's components that we very clearly understand, and I'm going to talk about some of the therapies that are targeting these components, as will Dr. Magnuson and Dr. Moritz. But there's other components where we're just starting to see the picture. And there's other components which we probably don't even understand yet. And it's going to require understanding this whole complex system of changes that happen after injury to really get good, strong recovery. And so we know that spinal cord injury involves a series of changes. You have the initial acute injury. This initiates a series of events that lead to changes within the cord that eventually resolve and you get a chronic injury. So what are these processes that happen as the injury progresses from acute to subacute to chronic? So let's look at this. Well, the initial injury is a primary injury, and it's restricted to a relatively small area. But what happens is the blood vessels break, blood comes in, you get swelling of the tissue, and you get this expansion of the injury site. This happens over the first few hours to days. You get the infiltration of inflammatory cells. You get the death of the tissue that's within this region as it expands. It can start to impact axons and neurons that were initially spared. And then a series of wound healing events take place where the injury site is walled off to try and contain this damage so that the tissue that is preserved can start to function again. So the temporally evolving nature of spinal cord injury means that we're going to require different therapeutic approaches for different times after injury. And the multicellular nature means that we're going to have to target different cells, probably at different times after injury as well. So how do we go about repairing the spinal cord? Well, early after injury, we want to try and reduce the initial damage. So obviously, prevention is best, but once the damage happens, what can we do? Well, frequently it requires surgical intervention to decompress the spinal cord and the administration of neuroprotective strategies, which suggest protecting neurons, but also involve protecting glia. We then need to think about how we can maximize the function of the spared circuitry, things like epidural stimulation and rehabilitation. And again, I think Chet Moritz is going to talk about some of the work that he's doing on these areas, as will David Magnuson. We want to think about how do we promote tissue repair? How can we alter the wound healing and inflammatory responses that normally happen to get less tissue damage and more repair? How do we reduce the environment that prevents the system from rewiring? And how do we enhance the ability of axons to grow? And lastly, we think about strategies to replace the cells that are lost using cellular therapies, including replacing the neurons that are lost and the glia that are lost. And so to do this in the lab, we use, for the most part, rodent contusion models of spinal cord injury as a field. My lab in particular, we primarily focus on the rat, although we've done some work with mice. And what you can see is the progression of these changes that happen within the injured spinal cord. So we're looking at longitudinal sections of the spinal cord. The white matter is located here. The gray matter is located here. This is more gray matter and more white matter. And all of this yellow is a hemorrhage that's happened and is present within the cord initially and after injury. As the injury progresses, you can see that the hemorrhage starts to resolve. At 1 week after injury, you can see the injury site is delineated here, and this whole region is filled with macrophages and inflammatory cells. And then over the progressive weeks to months, the injury site starts to clear out. You can see new tissue forming within the lesion, but ultimately you end up with this large cystic lesion with a spared rim of axons. And so I think we like to think that these models mimic human spinal cord injury very well, and for the most part they do, but rats are not humans and there are substantial differences. And I think another topic that Dr. Magnuson is going to talk about is some of those differences that exist in terms of the anatomy and the functional outcomes of rats and humans. So in my own lab, there's really three main areas of focus. One area is examining the cellular changes that occur after injury, and that how those cellular changes influence the development and recovery following injury. And so in collaboration with John Cave, while I was still at Burke, we looked at what happens if you modulate the response of astrocytes after injury, and if you delay that response of astrocytes, you exacerbate the lesion injury. More recently, with my colleagues at the Neural Stem Cell Institute, we began to look at single cell analysis to profile some of these changes that are happening after injury. Another area where we've been focusing for a long time is testing therapies to protect the spinal cord. And in these assays, we look at how well is the tissue spared after injury, and can we get improvements in function. We've done this through a series of studies, collaborations with Brett Langley while I was at Berkeley, looked at elevating levels of NAD within the cord. And more recently, we've collaborated with a drug company to start looking at a new drug that shows promising protective effects. The last area of interest in the lab has really been optimizing and testing cellular therapies. And I have a long history of this work from the time that I was a graduate student. And this is the area that I'm going to focus on a little bit more today. So a number of different cell types have been tested for spinal cord injury repair, and I'm going to talk some about the translational progress of these therapies. So these therapies include looking at fetal tissue grafts, looking at embryonic stem cells because they have the potential to differentiate into multiple cell types, using primary cells such as Schwann cells and olfactory and sheathing glia because of their ability in their innate locations to promote the ability of axons to grow. And more recently, technologies have been developed where you can reprogram cells so that you can differentiate adult cells into these various components and use those for repair. So really the first 30 years of research on cellular therapies was preclinical. It started with looking at peripheral nerve grafts and fetal tissue transplants, and then progressed to looking at more individual cell types. And it took a long time to do these studies because they were questions such as, what do these cells do following transplantation? If they have the capacity to become multiple cell types, do they differentiate into those appropriate cell types? How do they integrate into the spinal cord? Do they promote the ability of the tissue to repair itself? And so we were able to answer many of these questions with these approaches. And some of these therapies have since gone on to begin to be tested in humans. So if you look at clinicaltrials.gov, which is a resource for identifying all the clinical trials that are registered, currently I think there's 57 clinical trials on clinicaltrials.gov for spinal cord injury and transplants. And so this research has really progressed in the last 10 years from the very first transplantation trial registered by Geron, which has undergone a series of company changes. The latest version is Lineage Cell Therapeutics. But these trials continue to progress, and the results have started to be published. So we're starting to know, at least for Schwann cells and oligodendrocyte precursor cells, that these approaches are safe. Just last month, the results for the OPC safety studies were published on clinicaltrials.gov. And so we're starting to really have some progress in these areas. And while it's great that we're starting to see that these studies are safe, I think really what we want to know is, are they efficacious? And I think as we continue to move forward, we're going to find the answer to that question. But there are some challenges. One current limitation of cellular therapies is we tend to focus on a single cell type or a couple of cell types rather than the complex array of cell types that are present. And so we don't know, are these other cells required as part of a transplantation paradigm? And do we need to start including them? And I think as we move forward, we're going to start to see the answers to those questions. Another limitation of cellular therapies is that after injury, the environment differs from that found within the intact spinal cord. So in this paper by Wuhan et al., what they found is that there were 8 cell types within the uninjured spinal cord. And after injury, there was a shift in the proportion of cells within each of these clusters. In another recent paper by Milch and Choi coming out of Jay Lee's lab, they were able to identify 15 different cell types within the injured spinal cord, indicating that after injury, the environment is much more complex, and we still don't understand how these cells are impacting the transplants. In work that we've been doing with other investigators at the Neural Stem Cell Institute, we're finding that within a single cell type, there are changes in the proportion of cells. Some cells appear after injury, some cells shift their proportions after injury, and understanding how these shifts in cell types impact transplants will also be important. But we've made tremendous progress, and this progress that we have made is because of the ability of researchers to build on each other's work. So in the early 1980s, work coming out of Albert Aguayo's lab was the first to really show that the central nervous system had the capacity to regrow. Similarly, work from Paul Ryer's lab showed that if you put fetal tissue into the spinal cord, axons were able to go in, neurons from within the graft were able to send axons out into the spinal cord, so that these transplants were able to form relays. Other investigators then followed these initial studies to improve on their effects. Richard and Mary Bunge looked at Schwann cell transplants. They identified that Schwann cells were the principal component within the peripheral nervous system that allowed for the axons to grow in the peripheral nerve. And they showed that through a series of studies, when you added additional factors, you could get further improvement on the utility of these cells. Similarly, Dr. Bregman's lab in the 2000s showed that if you add growth factors to fetal tissue transplants, you could get improvements in function, something that wasn't initially achievable with the early transplants. And we've continued to learn about what happens in these transplants and modify them further. When I was a postdoc at the Miami project, we started using labeled cells for the first time, and we're able to see that within the first 7 days, there was a reduction in the number of cells after injury. We went on to show that these cells were dying within this time frame primarily by necrosis, but also by another cell death process called apoptosis. And we then began to target it, and we identified a calpain inhibitor that blocks necrosis that improved the survival of the cells. In my own lab, we then went on to show that we were able to narrow this window down to the first 2 to 3 days after injury. So this initial period of acute transplanted cell death occurs very early after injury. And we've gone on to target this early window of cell death. Mark Tazinski's lab has taken a different approach, along with a series of other investigators in the lab. They showed that if you transplant fetal tissue cells along with a matrix, series of 11 different factors in this initial experiment, that you can get robust regeneration out of the grafts. They've subsequently been able to narrow this down to 4 growth factors along with a matrix, and included in this is the calpain inhibitor. So you can see how the field has built upon each other's results to propel the field forward. But along the way, there are going to be dead ends. And with these dead ends, we're going to have to find different approaches. And so in the last few minutes I have left, I'll tell you about a dead end that we've encountered. And that is trying to target this early phase of death with a process called preconditioning. So preconditioning is one of the most protective effects known. It involves mildly injuring a cell, which results in the cell shifting the expression of molecules, which then allow it to survive when it's exposed to a subsequent severe injury. And in a number of transplantation models, this approach has been highly protective. It can be induced by a number of different initiating events, such as hypoxia, ischemia, heat shock, and the effects are then not restricted to the protection to that same induction. You can have a broader profile of protection. One of the ways this approach works is through the activation of transcription factors, which bind to DNA and then result in the expression of target genes. So instead of trying to target a single molecule, you now have groups of proteins that change their expression in response. Importantly, we can mimic this effect pharmacologically or by overexpression of growth factors or transcription factors directly. And so we started with an approach where we used the induction of hypoxic adaptations to increase the expression of target genes of a transcription factor called HIF1 alpha, or hypoxia-inducible factor alpha. And this is a transcription factor that's involved in cellular processes that we think are important in this initial phase of death, including oxygen transport and delivery, cell growth and fate, and changes in metabolism that are important for the survival of the cells. What we found is that if we overexpressed or we forced the expression of HIF in Schwann cells, and then we transplanted those cells, we could increase the survival of the transplanted cells. We then went on in a series of experiments to use pharmacological inhibitors of HIF, pharmacological inhibitors that elevated the activity of HIF. And through a series of experiments, we were able to identify pharmacological agents that promoted the stabilization and transcriptional activity of HIF. And then when we tested it in vitro, we were able to get an increase in the survival of the cells in these culture models. However, when we tested it in vivo, so we applied the pharmacological agents at the same doses and durations that were shown to be protective in vitro, we saw no protection in vivo. And we've done these experiments at least 3 independent times with similar results. And so really indicating that this approach for Schwann cells is not protective. So again, along the way, there's going to be some dead ends. It turns out that hypoxic preconditioning and induction of hypoxic adaptations in Schwann cells is a dead end, but at the same time, Mark Jasinski's lab has developed an approach with this combination of factors that is protective. And more recently, Giles Plant, using Schwann cells in collaboration with other investigators, have developed a biomaterial. And so we had focused on trying to not include any additional factors along with the cells, but it seems increasingly likely from the evidence from our lab, as well as the evidence from these other approaches, that including factors along with the cells is going to be an important component to enhancing the efficacy of cell transplants. So I hope what I've shown you is that there are areas that we understand what's happening after injury. There are areas that we partially know that are starting to become more clear. And there's areas that we don't yet know that are maybe just starting to emerge. And so as we continue to move forward, we will continue to clarify this picture of what is needed for recovery, and that will allow us to develop better therapies. And so I'd like to thank everybody for the time and for listening. I'd like to acknowledge that there was a lot of people that contributed to the studies that from my lab and from the work that I did before I had my own lab, as well as I tried to acknowledge the work from others in the field who have contributed to really moving this forward. So I'd like to thank the investigators at the Neural Stem Cell Institute, who I've been collaborating with since I've moved, as well as the members of my lab while I was at Berkeley, as well as other investigators at Berkeley who helped me along the way. And of course, it wouldn't be an IOSCIRS seminar if I didn't show the pets. And so here are my 2 cats, Maddie, who's clearly learning more about spinal cord injury, and Yip, who's doing what he does best, which is to sleep. And so with that, I will happily take any questions.
Thanks, Dr. Hill, and thank you for really an eloquent discussion of the complexity of the cells and the complexity of the problem. I think that's really important. I know in our work, a lot of times folks have a very maybe overly simplistic view of stem cells. Like get me some, why can't I get some stem cells in me and get some recovery? And I think you did a nice job of explaining that complexity in the anatomy of structure and function or connectivity. And also I wanted to point out I like your power failure metaphor, but wanted to add a little bit to it because oftentimes it seems to me like not only is it just a power failure, say in a city, but it's like a city on the moon. So far from the ability to get in there and do it, and then also understand, okay, well, what's going to happen in this environment, which we don't fully understand, right?
Yeah, it's really a challenge. Maybe it's more like trying to fix the Hubble microscope or the telescope.
Right, in orbit. So kind of along those lines, one of the first questions was from Franklin at NorCal SCI out on the West Coast. What are the top 3 obstacles that you encounter that keep you from significantly accelerating the advancement of your work?
Top 3. I think that there continues to be these areas that we just don't have enough information about. So I think that there's a real need to continue basic research of the changes that happen after injury. And sometimes this gets, categorized as descriptive, but understanding these components is really important. Another challenge is that these studies are really labor intensive and they frequently don't work. They don't work in the sense that we have really good ideas or what we think are really good ideas that end up the approach is wrong or the hypothesis is incorrect. And so we have to come up with new ideas and new hypotheses, but that's just part of doing science. Third, wow. I'll have to come back to that.
Okay. All right. Well, maybe a sort of related to your answer, particularly the first answer about information, another question from William, which I think is interesting because I know in some ways what you've talked about when you mentioned Tuszynski, there is a growing sort of look at, okay, how do these cells develop organically in the body through development, fetal and early childhood. So maybe you could comment a little more on that understanding. Where has that sort of tributary of the research informed back to everyone else?
So many of it seems to be quite similar. The ability of the cells to modulate the system changes over time. And so that's probably a big difference between earlier in development and in adulthood. In adults we know how to walk. Children don't know how to walk, right? They have to build the circuitry and adapt the circuitry and learn and consolidate that circuitry. So that's one fundamental difference between them, and we're still trying to understand how that happens and what cells might play a role in kind of setting up the system and consolidating it.
So does that also apply? I mean, a kind of offshoot question. You had talked at the tail end about, some of the growth factors or factors that are being added to the cells to, explore a better efficacy in the result. Is there an opportunity for the modulation more organically? So if you understood more about what each of the cells' roles were in that development, that the cells are delivering those factors in development, the cells are delivering those factors in normal function, so how much of that is understood so that you don't have to isolate them, but sort of more in the natural processes of the body to modulate factors?
So there's certainly very clear progressions of developmental cues. And, there's a whole aspect of biology that studies development and the progression of these cues. And I think that we've been able to take that information and apply it to culture systems. So the development of organoids that resemble, the major cellular components of the brain. Part of our ability to do that is understanding those basic developmental processes and the cues that are required. And so people are now using these organoids to try and understand how development happens and how cells interact. And that information is also being used in our ability to reprogram cells and push them down specific lineages.
Okay, great. Thank you. We're going to have to stop there with the questions, but I do want to remind you that we'll have more time with all three presenters at the end to continue the questions. So thank you for asking them. Maybe we'll get back to them later. Now we are going to move on to our next presenter, Dr. Chet Moritz. Hello, Chet.
Hi, Matthew. How are you?
Good to see you.
Good to see you as well.
Dr. Moritz comes to us from the University of Washington, where he holds, I believe, positions in 3 different departments. Is that right, Chet?
That's true.
He's an associate professor in the Division of Physiology and Biophysics, Department of Rehabilitation Medicine, and Electrical and Computer Engineering. How about that? And Dr. Moritz's lab is working both in animal models and in humans to develop treatments for paralysis using brain-computer interfaces and neurotechnology-like devices that Dr. Moritz is going to talk about today. Welcome.
Awesome. Thank you, Matthew. And thank you, Dr. Caitlin Hill, for such a wonderful introduction. It'll allow me to really just jump straight to some of the work that we're doing in our lab and in collaboration with other labs around the country and tell you about some pretty exciting research. You probably heard about some of it involving both epidural stimulation as well as transcutaneous or skin surface stimulation, which is what I'm going to focus on today. And I'm excited to tell you about the improvements we've seen for people with cervical spinal cord injuries, all at least 1 year post-injury, so we call that chronic injury. And we'll talk about the improvements we've seen in hand function, now across 7 individuals. We'll talk about some ongoing studies looking at walking function in about 4 individuals. And then we'll talk about some other stuff. And I put this in as a tip of the hat to Dr. David Magnuson, who will go next. And I can't let David be less formal than I am. So, well, he probably still will be, but I had to put this in as well. But the other stuff is pretty exciting. It's bladder function, it's bowel function, it's thermoregulation, and not everyone gets all the benefits, heart rate, blood pressure, but it seems like each person gets something. So it's kind of like a grab bag at a party. Everybody reaches in and gets some random other stuff at the end. So we'll spend a few slides at the end highlighting that. All right, let's start with hand function because that's the most developed story we have. We have a couple papers published on this. The first one there is referenced at the bottom. That's just a case study. We'll highlight progress by an individual named Joe. He's okay if we share his name. And anytime I use a name today, the participant is given permission to share their name and their identity. This is Jessie here. She's also happy to be known publicly as part of our study. And we'll talk about some improvements we've seen. First study on 1 person, next one on 6 people, and now actually part of a multi-site clinical trial with the company Onward, where we're aiming to enroll about 60 people across about 12 centers in North America and Europe. Happy to answer questions about any of that when we get to the end of the time today. So what's the secret sauce here? If you haven't heard about this yet, it's the high frequency stimulation. The 10 kilohertz stimulation allows us to pass about 5 times more current through the skin for the same level of tingling and prickling sensation that you get on the skin. That high frequency basically anesthetizes the pain fibers in your skin, lets us send more energy towards your spinal cord. And where do we think that energy is going? We think it's going to the dorsal roots, to the sensory roots on that same side, that posterior side of your spinal cord, through that, through synapses, exciting the motor neurons, getting them closer to threshold, ready to cause a movement. But we always stimulate below threshold. So the user chooses when they want to move, when they want to stop. The stimulation just makes it easier. So what does it look like? Here's our first participant. His name was Joe. He was about 3 years after a C3 incomplete injury, central cord syndrome. He was Asia D. And he improved quite a bit. We applied the stimulation early in this study in the orange shading here for about a month. So we have a couple baseline visits here. If you can see my pointer on this screen, I'm not sure if you can actually, maybe get a real pointer. We're quite sure what shows up on each different platform. Here we go. So 3 baseline visits. Then we stimulated for a month. Stimulation off in blue, on in orange. He's a little stronger each time with the stimulation on, but even with the stimulation off, he does pretty well. Then we do standard physical therapy here as a control, and we'll start with that control in all the subsequent subjects. A little more stimulation just to see if we can get an additional boost. And then this is the most exciting part of this graph and some of the others that I'll show you. The most exciting to me is that for at least 3 months after the stimulation ended, Joe maintained all of that function, all the strength in this particular graph, but also dexterous finger function, as I'll show you in a minute. So even though stimulation was required about 3 days a week, 3 to 5 days a week for a month or two, it's not required permanently, right? It causes some kind of neuroplasticity to change and recovery to be prolonged. And I should just mention this is a collaboration with Dr. Fabien Anici, who's a physiatrist and a PhD researcher, and Dr. Christoph Hofstetter, he's a neurosurgeon, as well as a spinal cord injury researcher. So check both of them out. They're both doing tremendous work on their own in addition to this collaboration. All right, let's see some movies because it's more fun to watch movies than watch graphs. So here's Joe, and this is his performance at the 9-hole peg test. So picking up these little pegs and moving them over into the other holes. This is what it looks like at baseline. He's unable to grasp any peg. The 1 or 2 that are out of the board got caught between his fingers and fell out, but he can't pick up the pegs. He can't get his fingers and thumb together. This is after 4 weeks of stimulation, so near the end of his treatment. Notably, this is with the stimulation off. So this is the persistent benefit that he saw from that month of stimulation. He's slow, he's deliberate, he's got a little tremor, he's got a little clonus, but he's able to grab and transport those pegs. We fast forward there. He did do it in 84 seconds. We just didn't want to make you sit through the whole minute and 15 seconds there. Interestingly, the very next day, if we turn stimulation on, he's about twice as fast. So the stimulation still helps him. We think of the stimulation as a gain knob, as a volume knob on the spinal cord, helps it listen to the signals coming down from the brain. Maybe a hearing aid for the spinal cord is a good analogy. So it still helps, but it's not required. And he was able to get back to his daily function, feeding himself for the first time with utensils since his injury, living more independently. So that was really exciting. It motivated us to design a study with 6 individuals where we did a crossover design and we looked at just training alone in blue, then the training plus stimulation. We repeated that twice. We randomized the second block. Really what we're trying to control for here is, do people just get better over time? Is it just the therapy that they're getting, or is there something causative about the stimulation? And I think what you'll see in the data coming up is it really is the stimulation that causes the improvement. The training alone, either no change or just a really subtle change. So same 10 kilohertz waveform using the prototype device. This was made by Neuro Recovery Technologies at the time out of UCLA, one of Reggie Edgerton's startups. It's now merged with Onward. It's the same stimulator that's being used in the Onward trial. So all this is consistent if you're hearing about any of the latest news. All right, what does this look like? I'm just going to play you the little summary video that the National Science Foundation made for us, because I think it just summarizes how we do the stimulation and the kind of functional and quality of life improvements that people have seen. Then we'll break it down and we'll look at the individual videos. But this is a nice overview of the studies just to get you oriented. I do need to share my audio here, and I'll mute myself and we'll play this one.
University of Washington have demonstrated a way without surgery to supercharge the progress of regaining hand movement. External skin patches that deliver electrical pulses applied to the back of the neck over the damaged areas of the spine. The pulses don't cause a muscle to move, but get it ready. Prime it to move when the patient wants it to. 6 people with spinal cord injuries were recruited for the 5-month study. Combinations of physical therapy with this external stimulation and 3 months follow-up. Results? Nothing short of amazing. During stimulation, those with no hand movement could move their hands, showing measurable force between thumb and forefinger. Others went from no use of their hands in daily tasks to being able to play a guitar or use a paintbrush on canvas. These effects remain even after 6 months, as long as could be measured, with no additional stimulation, helping patients regain physical movement and independence.
Awesome. So hopefully you could just see the impact there and a couple of short clips, but let's break it down and let's look at the actual data. So I always get the question, well, that's nice. People with incomplete injuries, these are Asia C's and D's mostly. What about Asia A and B? Have you worked with people like that? And indeed we have worked with 2 people who are Asia B. My colleagues at Craig have worked with people who are Asia A. And so far it's basically working in almost everyone that we have enrolled. So here's an individual who is Asia B. He had no movement at all of his fingers and thumb on either hand. So he's paralyzed from the wrist down, C5, Asia B. And we tested him over and over again, pinching this little pinch force that's shown here in the upper right, pinch force meter. Couldn't move, couldn't register any pinch force throughout a month of baseline and a month of physical therapy only. And it was only after about 2 to 3 weeks of stimulation that first his right hand in blue and then his left hand in red started to move for the first time since his injury. Been about 1.5 years since injury. And he's able to produce measurable pinch force. Another month of stimulation coupled with training, and that force approximately doubles again. Still weak, still less than 4 newtons. So that alone, probably not functional, but coupled with his really robust tenodesis grip, which he'd already been using, he's able to drive his car with the modified hand controls. He's able to even start working on inserting a urethral catheter by himself. So lots of independence and dignity improvements there. Again, my favorite part of all these graphs, we got 6 months of follow-up on this particular individual thing. We thank him for coming back the 6-month mark, even though it wasn't required in the study. And he never drops below the level of force where he left the physical therapy part of the study. So again, looks like a persistent improvement that doesn't require stimulation to maintain that improvement. All right, let's see some more videos. This is back to the individual named John. He was 12 years post-injury when he joined the study. He was 13 years post towards the end. So you'll see both those numbers on some of these slides. And let's just look at what some people see early in stimulation. So this is John's function without the stimulator. He'd done 10 out of 12 days of the training alone, but hadn't been stimulated yet. You can see his right hand can move, but it's functionally so weak, he can't even pick up these ping pong balls. These are light, hollow plastic balls. They just fall back on the table. This is within the first few minutes of stimulation on the very first day. He's now able to grab and release those balls quite easily. So that's a quick change. Not everyone improves the very first day. Some people take a week or 2 or 3 to start to see improvements. John improved right away, which was a lot of fun to see. Here's another example. This is now on the second day of stimulation, but the stimulator is off during this first clip. So he's graduated from those ping pong balls, you see him in the background, now to these small wooden beads about the size and weight of a marble. He's kind of can get a hold of them, but he can't keep a hold of them until he drops them in the bucket. And then here's where we turn the stimulator on just a few minutes later.
Oh yeah, that's crazy. Wow. Oops, I'll slow it down a little bit. I'm acting like I'm a pro at this now. I'm just holding it and open it. Wow.
Helping, yes.
Oh,
And this
man.
is 45. Let's go with this one if you can tolerate.
God. This is worth any kind of pain. This is amazing.
It's really amazing.
Wow. Like the other ones, like if I can get them, I'm like, they're just barely slipping out of my fingers. I can barely do it, but that's like a pinch.
So
That was really cool.
Yes.
It's even crushing. Wow, that is really, wow.
And then this next video clip was about 3 months after the study, but with the stimulator off. So again, his sustained benefit, he kept improving during stimulation, he kept improving even afterwards, using his hands in everyday tasks. And that's a little friendship bracelet bead. I can't even pick those up. I have to have my 4-year-old daughter pick those up off the table for me. They're just tiny. So really exciting to see John's improvements, but it wasn't just limited to John or Jesse, as you saw in the movie. Other people improved as well, including people with a lot of spasticity, with strength, but a lot of tone, a lot of spasticity. And so this video shows a woman who's about 2.5 years after an Asia C5 injury. Her challenge is not strength, but it's opening her fingers. You'll see her stretching there, trying to get her fingers open enough to grab one of these 1-inch blocks. It's a box and blocks test. You've probably done this. You're supposed to pick up the block and throw it over the bar. She gets creative and kind of scoops it, the side of her thumb, but she can't grasp. And 12 days of physical therapy alone didn't really change at all. If anything, it just taught her to give up and not really try to scoop. So therapy alone didn't help. But when we turned the stimulator on, by the 3rd day, her spasticity, her tone had started to reduce, and she's able to grab and release the blocks when she wants to. A few days later, she's got even more control over this grasp and release. Same size block, but she's able to just very delicately stack them. So look at that control. Remember, her fingers were literally stuck together before. Now they're opening by 1 inch. A few more days, she's able to open them even farther. Well, that's exciting to see. You'll see here by the 18th day of stimulation, so near the end of stimulation, now she's grabbing things that are closer to 3 inches, these wooden blocks that are closer to 3 inches. So remember, again, her fingers and thumb were stuck together, couldn't open even to grab that small block. Now she's grabbing blocks that are about 3 times the size. So that's really exciting to see. All right, one of the last boring data graphs I'm going to show you. Just want to show you that it worked in all 6 participants in the study. Each colored line is participant 1 through 6. They start out, they get a little bit of benefit from training alone. That's the blue bar. The average is the bar graph. Each individual participant is a dot with a line connecting them. But while they might go up by, a couple newtons of force during training alone in their pinch force, that's what's shown here, they go up by more like, 10 to 24 newtons. When the stimulator is running. So often like a 7-fold difference in force when the stimulator is running. So really impressive, really functional. This is a measure of strength of all 10 muscles in each arm, part of the grasp test. It's part of those manual muscle tests, plus some extra muscles. Again, you might gain a few points on that test, but you gain a lot more. You gain maybe even up to 25 points out of 100. The scale only goes up to 100, so you're getting about a quarter stronger. Prehension, again, just a measure of those. It's the 9-hole peg test and a few other things, keys and locks, nuts and bolts. putting a coin in a slot, all these things, getting much better during stimulation, really staying about the same during training alone. That's what these graphs mean. All right, that was hand function. We're pretty excited about that, and we're pretty excited to be part of the multi-site clinical trial. And actually, Caitlin, go ahead and pop that link in the chat now. We are recruiting for both this study as well as 2 more follow-up studies. We just got funded on hand function, funded by the Nielsen Foundation, funded by the Department of Defense. So do please contact us if you're anywhere near Seattle or you're able to relocate to Seattle. And even if you're not, contact us. We can put you in touch with the other sites that are part of the Onward trial. There's about, like I said, 12 sites in North America, US and Canada, as well as Europe. So happy to connect you there. Not going to spend as much time on the next 2 sections, but I do want to just tell you that this same transcutaneous stimulation seems to help people improve their walking ability as well. We've completed the study on 2 individuals. We've got 2 more ongoing. So I'll just briefly summarize that, show you just a couple examples, and then towards the end, we'll talk about that other stuff. So stay tuned for that, because we've got some great videos of how the stimulation, coupled with a lot of hard work, has changed people's quality of life. Actually got some videos out at people's homes and things to highlight that. All right, so here's walking. What did we do here that's unique? Well, we used the same 10 kilohertz waveform. We still applied it to the back of the neck, because all of our participants so far have cervical injuries. They have injuries up in their neck. But we also added 2 more electrodes over the lumbosacral enlargement, L1 and T11 bony landmarks. Let us target that central pattern generator in the lower back. And then we start by training standing and balance. So standing here in our safety harness. This harness can reduce gravity if we need to. Toss balloons back and forth, for example. This is Soshi Samjima. He's a new PhD, actually on his way to do a postdoc with Andrei Karasakov at UBC at ICORD. I'm just thrilled to be part of his training. He's got a bright future, I think, so keep an eye out for Soshi. Then Soshi started walking on that treadmill, again, with some body weight support at first, controlling the speed of the treadmill based on how much the individual leaned forward. This was Joe again. Just anecdotally, the reason Joe's in this study, when he was doing even our hand study, he was telling us it was easier to take steps with his caregiver helping him balance. When he was doing transfers, maybe getting into the shower. So that was partly what motivated us here to work with people even who have cervical injuries. Then we start over ground training. Again, we've got a walker there for Joe. His hands are still a little weak, so we got those modified walker with the elbow supports. And that's where we started the study. So she was holding on to him. We call that full contact guard. Joe was leaning on that walker for a lot of the weight support, or leaning on the body weight support harness. But by the end of the study, only after about 2 months of stimulation, he was taking steps with just a single quad cane, a single 4-point cane. And so she just standing nearby in case he were to fall. So walking, not in the community like that, but taking steps in the lab. So we think that's a big change. And of course, his walking distance and speed all improved about 3-fold. And we'll show you some figures that summarize that. And then we'll show you one video at the end from our other participant. Again, kind of boring graphs. I don't want to spend too much time on them, but just notice the difference. The blue shading is the training only. So just physical therapy, locomotor training. We always do an equal amount of that to make sure that's not the reason. The yellow shading is when the stimulator is applied, and the same with the same training. And so if you notice the difference in the slopes of these lines, sure, he gets better with locomotor training. He hadn't done that in several years. Gets better at a much faster rate and to a much higher level when the stimulator is running. Kind of plateaus here for locomotor training and improves again with the stimulation. And again, maintains almost all those gains for 3 months with no more training, no more stimulation. Same story for our second participant. Is improving very gradually with locomotor training, so that's good. But when the stimulator turns on, that's when the slope of that line really changes. And here we have 2 months of follow-up, and then COVID happened, so we don't have that last data point. But in communicating with him, he's maintained all those same gains. If we just sum up that data across both these participants, so all the gains in yellow and all the gains in blue or purple, we get that training effect right down here, right? It's modest, you know, improving about 25 meters or so in a 6-minute walk test. But when the stimulator is running, that's when we get 3 times more improvement, up to over 100, almost 120 meters improvement total, right, if we sum the two with stimulation. And that maintained to 2 to 3 months of follow-up almost at that same level. And of course, the minimal clinically important difference, if anybody's into stats, is right down here at the dashed line. So the training alone, barely clinically important. The stimulation about 3 times greater. So we're excited about those changes. All right, it's easier to see it in a video. This is our second participant. His name's Steve. He's okay if we share that. He's 4 years out from a C6 Asia D injury, again, central cord injury. Got a little better function in his legs, actually able to ambulate in the community with a crutch or a pair of crutches. And you'll see his walking function here. And for these videos, we're going to support 21% of his body weight throughout, even though he doesn't need that by the end. And we're going to set the treadmill speed to his maximum speed. At baseline, he was about 0.6 meters per second. And he needed a physical therapist. That's when our PT's Emily moving his leg forward, his right leg forward with every step. They needed 2 months of locomotor training. And he no longer needed assistance for that leg. So that, again, that's progress. And he's walking just a little faster, 0.7 meters per second. So 0.1 meters per second faster. Still just going to support 21%, just to keep it consistent for this particular comparison. Then he does 2 months of stimulation with training, and this is the result at the end. Now he's walking 2 meters per second. It's almost a run. You can see he's not getting into the air there, but he's moving very quickly. Still supporting 21%. We don't have to. At this point, he's walking without his crutch around his house. He's moving around the kitchen. He's washing his big pickup truck, getting up on ladders. He's able to maintain his thermal regulation even when it's cold. He moved here from Texas, so the winters were tough here in Seattle. Just big changes there. And that's really how I want to finish. I want to finish with the other stuff. And again, this is the grab bag. Not everyone gets other stuff. Not everyone gets everything in the other stuff bag. That's definitely true. But it seems like almost everyone gets one thing. And the list is long here, and I'll go through a couple specific examples. I know this is wordy, but I'm just going to read through it just so you realize how many people this happens for and what kind of other stuff is out there. So bladder and bowel function. 1 or 2 people, one person, in this case, discontinued the need for intermittent catheterization over the course of 2 of our studies and continues to not need that. Is able to void spontaneously now about 3 years out from the study. Again, we can't take all the credit. There's probably something else going on, but all the improvements in bladder function happen during the periods of stimulation. We've had 2 people now significantly reduce the times for their bowel program by about half, even to 1/3 of the time it took them originally, more regular and less time to manage their bowels. We've had 1 person have heart rate and subsequently blood pressure return to normal from a very low resting heart rate and low perfusion, such that when the individual sat up in the morning, they felt lightheaded and dizzy, couldn't concentrate, couldn't focus. During the study, that heart rate came from the low 40s at rest to the mid 60s. It's been there ever since. Obviously, it modulates around that level, but able to feel much more alert, much more productive in daily life. And 1 individual start to sweat again below the injury. Different individuals start to feel sensation, hot and cold water on their skin. Had several individuals improve their cutaneous sensation. For example, starting at, say, the C4 dermatome, so about the collarbone, everything below that was either absent or altered, so it felt numb or tingly. After the stimulation, full 10 segments down to T10 normal sensation. Didn't completely last, rolled back to about T4, but still added about 4 or 5 segments of normal sensation. And then at the end, and this is what I want to just spend my last few minutes on, a couple individuals returning to the hobbies that they enjoy, like playing the guitar, oil painting, and you saw maybe some of that in the first video, but I wanted to show a couple more videos to kind of, even just from the last few weeks, to sort of drive home the point that some of these studies can change people's quality of life for many years afterwards. So finish with that. It's kind of a fun little way to top it up. So this first video is really fun. It just highlights the process by which stimulation makes people better. So it's John again, 12 years post-injury, 13 now by the time we took these videos. And John talks about how he had to work really hard while the stimulator was running. And that's an important point to emphasize. The stimulator is not magic. The stimulator gets your motor neurons closer to threshold, and you have to work really hard to turn them on with your brain, and it allows people who can't turn their muscles on to turn them on when the stimulator's running. So it's powerful, but it's not magic. You still have to practice, you have to work, you have to train, to put a lot of energy into the rehab part, as I'm sure all of you know, in order to get those long-term improvements. So this is fun because it's John talking about how he relearned how to play the guitar. He hadn't played the guitar in the past 12 years. Just he could move one of his hands, not the other one. Just wasn't any fun. But you'll see in this video, I think, that how he talks about the hours of practice that he put in to playing the guitar, both during the time where we were stimulating him. And then he said for about 4 hours after each stimulation session, he felt, quote, potentiated. I think he got that term from us in the lab, and that he felt more function for about the 4 hours afterwards. So he would use that time to practice the guitar, practice using his fingers.
So you start like pull what you can use into it.
Yo, So what did your thumb do before on the right side?
Nothing.
Right. You just wouldn't just.
It just got in the way.
Yeah.
Yeah, just like and I focus and work at it for an hour. You know, it's like we can strum it.
Right.
And we can get that movement going. It's like we can't do it right now. So I'll just sit there for like a half hour and just like pulling it down.
That's why that particular movement's gotten so
That.
much better.
Just do it over and over and over until it starts to get a little better. Yeah. And that's how we even got here at all. It's like just would do this like just over and over until I could like, get it. It started like I couldn't even make the sounds. I couldn't push it down hard enough. I start to get it, and I start to do other ones, and pretty soon the chords start to come together. And I started to be able to play them on the neck, which that's like all those fingers like working together.
And this is 6 months later with no more stimulation. So that's kind of fun. It's fun for us to see how much pleasure John gets out of playing the guitar. It was really fun when we checked in with him just a few weeks ago to look at the improvement that he's made from his first chord, which he sent us this little video when he made his first chord during the study. You can see his hands there, lots of tone. He's got to stretch them out. You might hear the audio here, just sort of a couple of strums. And it doesn't look like much, but John was thrilled when that happened, right? So he couldn't even do that before the study. And now look at him. He's playing lead guitar in a garage band in Seattle. So if you know anything about the history of grunge music, this is your preview of the next Nirvana. And that's awesome. And it goes on for a while. We can play it during the Q&A if we have time. It's a good little jam. And then I got the chance to visit John at his bamboo farm as well. This is even just a week ago. So he's a couple years out from our study now. He's actually participating in one of our walking studies. But look at this. He's got enough hand function to shovel potting soil into a pot. Again, you can see his hands aren't perfect, but they're strong. He's out there working 6 to 8 hours a day. Everyone seems to want bamboo during the pandemic to, shield their neighbors as they're all working from home and living at home. So he's got an incredible amount of business this year. He fills the pots, and then he uses his wheelchair as a tractor, and he delivers. These are like 40 or 50 pound pots by the time they have all the soil in them. He delivers them out to people's cars on the sidewalk. So again, that hand strength to move that wheelchair, just the stamina. He says he just feels more alert and more active, more awake. All these things we can't quantify other than his heart rate used to be in the low 40s, and now it's high 60s and even over 100 when he's exercising. So that ability to control his temperature, he can sweat now while he's outside. He couldn't do that before. And so just, it can be life-changing for some people, right? If they put in the effort to get better, and that's what we're learning. So just going to wrap up here, hopefully save a few minutes for questions. We talked about hand function first. We've seen in all but one individual who withdrew from the study before we got a chance to finish with him. So we're now on 6, 7, 8, 9 people, 9 out of 10, I guess we could say. And I'm still not sure about that 10th one. If he'd stuck with us longer, we might have seen improvements. Have improved hand function, and everyone that we've measured for follow-up has sustained for at least 3 months. That's how the study was designed, and we get up to 6 months if we're lucky. So, and then a little bit on walking. It's early days for us, just 2 people complete, but we're seeing that walking speed certainly increases, independence, ability to balance, move around the kitchen, wash your truck, play with your grandkids, all that is changing. And then for some people, again, I don't want to overpromise it because not everyone gets bladder and bowel improvements from this. Not everyone gets a normalized heart rate. Not everyone starts to sweat again. But somebody gets all of these things, and sometimes more than one person. So with that, I just want to thank the team. I don't have any pet photos, but I have my lab. I don't know if that counts. There's about 15, 16 of us at the moment, really passionate about spinal cord injury research. We're going to include all their names here and then all the funding that's supported us over the last few years at the bottom. So thanks for your attention. If I saved any time, we'll do questions. Otherwise, happy to do them at the end of all the talks.
Thanks, Chet. Pretty cool stuff. Appreciate that. I have a quick question before we get on to, there's a couple of questions here, but the first one is, put this in the context of, there are some companies out there moving to clinical trial right now for devices like this. Where does this fit into the device you used, the technology you used? Where's the intersection between that and obviously what people are going to be hearing about Onward or SpineX? Let us know.
Exactly. Yeah, it's all tightly interrelated. So the device that we use for all this data that I showed you today was the predecessor to the Onward device. So it's developed by Neurorecovery Technologies. Onward, which was called GTX Medical at the time, merged with Neurorecovery, acquired all that technology, all that IP. And I'm one of the co-PIs, one of the two co-PIs, along with Dr. Eddie Field-Bote at Shepherd, that are leading the multi-site clinical trial for Onward. So all totally related. That recruitment link that we hopefully popped up for you, that'll take you to my website. Contact us, we can connect you to the Onward site. So we're all working together. I'm also working with Parag Gad at SpineX. He's providing stimulators for a parallel set of studies. I can't tell you too much about the indications, but we're working with Parag as well. He's been a great collaborator with us over the years. Everybody's kind of connected, right? It all started in Reggie's lab. Gregor Cortine trained there. He started his own company in Switzerland. Reggie and Parag started Neurorecovery, then spun out SpineX. Everybody's talking, everybody's working together. So I think it's a really bright future.
Great. Yeah, there's a few questions here. So one of the first ones from John Chernesky: do you think the effective spinal stim differs between those with central cord system syndrome?
Sorry to interrupt, Matthew. Sorry. I'm just going to say we do have one more speaker, so we'll keep to one question if that's okay. And then we'll do big Q&A at the end. So make sure to get time for everybody's questions at the end. Thanks.
Okay, thanks. No, that's good. We'll stick with that one. There's several good questions we'd love to get to, but we'll save those for later. John, versus anterior injuries. Any thoughts about that?
Yeah, thanks for asking, John. This slide doesn't totally answer your question, but I can tell you, I don't think I marked it on here. Yeah, the stars are central cord injuries. So we have 1, 2, 3 people who are ASIA-D central cord. You saw at least 2 of them in the videos that I just showed. They definitely improve. So central cord is not a problem. I can't tell you about anterior damage. I think anterior horn, if the motor neurons are damaged, we do worry about that. We exclude people presently who have frank loss of muscle in very specific muscles, not just general atrophy, but if it's clear the lower motor neuron has been damaged, we're not sure we can help that yet. We're looking forward to trying that once we're through the multi-site clinical trial. Once we get FDA approval, we can circle back and test that on lower motor neuron injuries. That's the one I'm not sure is going to work so well. But so far, no difference between central cord and regular injuries, if that's a term.
Regular injuries. Okay. Well, thanks, Chet. We're going to move on to Dr. Magnuson. Welcome, David.
David, can you go ahead and share your slides, please?
So while they're doing that, I'm going to give an introduction to Dr. Magnuson, who comes to us from the University of Louisville. He's a founding member of the Kentucky Spinal Cord Injury Research Center and a Friends for Michael Professor of Neurological Surgery. His area of focus is on the central pattern generator, spinal circuitry before and after injury, rehabilitation and exercise, and especially inactivity and immobility and how that affects the trajectory of the injury. He has a PhD in neuroscience from UBC in Vancouver. Also, like Dr. Hill, kind of went all over the place, did some postdoctoral work in London and Ottawa and Manitoba, and now he's at Louisville. Welcome, Dr. Magnuson.
Matthew, thank you so much. This is a great opportunity. I'm going to apologize for my voice. I have my own little issue with paralysis, which is my left vocal cord, but we're hopefully going to get through this. it is a really exciting time to be in spinal cord injury research. The work I'm going to talk about today was primarily funded by the NIH and by the Kentucky Spinal Cord and Head Injury Research Trust. And I'm going to be surprisingly formal, Chet, just in case you were wondering. So I'd like to, so one of the reasons why I'm in spinal cord injury research is because I love the way that animals move. And I grew up riding a horse and totally was enthralled by how this animal with me on top of it could do the amazing things it could do. And the spinal cord is more than just a relay station. It's more than just a bunch of wires, although I do really appreciate the power grid analogy, Caitlin. But really the spinal cord is computing power, it's brain power that's been offloaded from the coconut, from the head, down into the parts of the body where that circuitry can be closer to the action. So in the Magnuson lab, we have this hypothesis. Rats and humans are rear wheel drive. We only study rats, but we like to include humans. The brain says go, and the spinal cord decides how, and then instructs the rest of the system what to do. So we're going to look at some things with respect to that hypothesis by first talking about the central pattern generator. And both Caitlin and Chet introduced this a little bit, Chet in particular. The central pattern generator is circuitry in the spinal cord that is able to generate a pretty decent locomotor pattern, even when it's completely isolated from descending input and from afferent input. So this circuitry, when given an appropriate amount of excitation, can generate a pretty decent locomotor-like pattern. And this is in vivo, actually in the animal, or ex vivo, if you take it out of the animal. And one of the crazy preparations that I did as a junior faculty member was an isolated set of rat pup hind limbs that were still attached to the spinal cord. And if you stimulate that spinal cord appropriately, sort of like epidural stimulation, or apply a particular set of drugs to tickle that circuitry, those hind limbs would step. And it's an amazing thing to see, and it shows the power of this central pattern generating circuitry to generate locomotor output. So one of the things I did many years ago was a really kind of crude experiment to try to figure out where the central pattern generator is. And other people had used other more sophisticated techniques. But what we did in my lab is we used an excitotoxin called canic acid, and then also a contusion injury. And we moved these excitotoxic or contusion injuries up and down the cord in the thoracic region and down into the lumbar region. And we discovered that the area of gray matter, in other words, that part that Caitlin was talking about with the neurons and glia, not the white matter, but the area of gray matter where we could get the largest deficit with the smallest lesion was when that lesion was centered on L1 and L2. So we believe that L1 and L2 holds the pattern generating circuitry that's most important for locomotion. Now, the entire spinal cord has rhythmic elements. That's what these little REs mean. And if you cut a piece of spinal cord out of a neonatal rat and you put it in a dish, you can actually get a pattern, an alternating pattern. But that doesn't mean that it's actually the locomotor pattern. So we think that these segments, L1 and L2, are actually the leaders with respect to the central pattern generator and for locomotion. So what is a central pattern generator? And my colleagues at Drexel will probably roll their eyes at the way I describe this. But anyway, it's basically groups of excitatory neurons, A and B, interconnected by groups of inhibitory neurons, A1 and B1. And these little black dots are inhibitory synapses. These white dots represent excitatory synapses. And so that when you excite this set of neurons, either A fires or B fires, because when A is firing, A1 inhibitory interneurons are inhibiting B. And when B is firing, B1 inhibitory interneurons are inhibiting A. So what this coupled oscillator, is the technical term for it, can do is take a constant input of excitation, sort of what might be delivered by epidural or transcutaneous stimulation, and turn it into a pattern of activity. And these vertical lines just represent action potentials in groups of neurons. So it can turn a tonic firing rate into a rhythmic firing rate that, if those neurons are actually output to flexor motor neurons and extensor motor neurons, can generate a basic flexor and extensor pattern. So this coupled oscillator in this case is not A and B, but is flexors and extensors. When the flexors are active, the extensors are inhibited. When the extensors are active, the flexors are inhibited. Now, we have bifunctional muscles in our limbs and other complexities, so it's definitely more complex than this. But this is basically the building block that the spinal cord circuitry is made of. And many, many years ago, this was called the half center. Not exactly sure why, but each limb, each quadrant of a quadruped, or for that matter, of the human, because we swing our arms when we walk, each quadrant has one of these half centers. And you can build these quadrants into circuitry that can take care of the idea of locomotion. So here's a flexor and extensor quadrant, half center, dealing with the right forelimb and with the left forelimb, and over here the left hind limb and the right hind limb. So these half centers, these key bits of circuitry for the locomotion output, locomotor output of the spinal cord, are interconnected by commissural interneurons locally. And that's a fancy name for just neurons that cross from one side of the spinal cord to the other, and long propriospinal neurons that connect the lumbar enlargement down here that handles the hind limbs or legs with the cervical enlargement up here that handles the forelimbs or our arms, and really importantly, vice versa. Information is being sent from the circuitry that drives our hind limbs up to the forelimbs so they know what to do. And information is being sent from the forelimbs down to the hind limbs so that the hind limbs know what's going on. Now, our hypothesis stated that we are rear-wheel drive. Therefore, this hind limb set of half centers is we think as being the most important for the system with respect to locomotion, not volitional movement like Chet was showing with reach and grasp, but patterned, repetitive things that we can do in kind of an automated way. So this circuitry gets reconfigured for different gaits. With our animals, we look at walk, trot, gallop, and bound. For us, it would be walk, jog, run, or skipping and jumping, depending on what we're doing. So this basic circuitry is really important. And how can we start to understand this circuitry? And we've taken an approach that I like to call reverse engineering, which means using techniques that can turn off some of these neurons and see what happens to the system. So the first set of neurons we're going to turn off are neurons that are represented by these blue arrows in the ascending direction that go from the lumbar circuitry to the cervical circuitry to tell the forelimb circuitry what's going on. Oh, I skipped something. Our hypothesis, the brain says go and the spinal cord decides how. We think that, at least for our rats, and we think probably also for humans, because let's face it, humans are rear-wheel drive, we're bipedal, walking with our legs. We think that most important signal comes down here to the lumbar set of half centers and then gets distributed by these long propriospinal neurons that I'm talking about from the lumbar circuitry up to the forelimb circuitry and back again. So we're going to try to reverse engineer this system by shutting off some of these neurons. And the first neurons we're going to look at are these long blue ones, and they're represented in this more anatomical circuit here. The cell bodies are down here at L1 and L2, and the axons go up in the lateral white matter, right near the edge of their white matter, right at the very outer edge. And that's really important because that white matter is spared after many incomplete injuries, and they terminate in the C5 to C8 intermediate gray matter, where we believe the half centers are for the upper extremities or arms. So we use this 2-virus system that was developed by Tadashi Issa. And when we inject these 2 viruses into near the cell bodies and near the terminals, and I'm not going to get into the technical things. In fact, I don't understand them myself, but I love what they can do. We can actually shut these neurons off. It takes a few days to turn them off, and then we can turn them back on again just by feeding the animal some doxycycline in their feed and then taking it away. And we can shut these neurons off. And here are these red guys showing what these neurons do. So they connect the lumbar circuitry with the cervical circuitry. By the way, this is a collaboration with Scott Whittemore, and the students primarily involved were Amanda Pokratsky, who's now doing a postdoc over with Rob Brownstone in London, Courtney Shepard, who's doing a postdoc at Children's in Washington, Brandon Brown, and our collaborators at Drexel University that help us with the modeling. Well, don't help us. They do the modeling. Are Simon Danner and Ilya Ryback. So when we shut off these long ascending propriospinal neurons, we see something very interesting. But the first thing we need to do is to look at how our rats step. And now my video is not working, which is really unfortunate.
Would it help if you turned off the laser pointer, maybe?
Well, man, oh man. This is really a bummer.
Yeah, I think if you can turn your laser pointer off, you might be able to get to the play button. Looks like, oh, Command and Spacebar or Control L. Thank you, chat, for giving us the shortcuts.
So Command and Spacebar gives me that.
Okay, so how about Control L?
Okay.
No.
There it is. There it is.
There we go. Thank you, chat. All right.
I don't know what I did, but oh no, it went away. Okay, so this is a rat walking normally, and you can see that we have beautiful alternation of the forelimbs and hind limbs. And when a pair of limbs is alternating, we say that it has a phase of 0.5 down here. And when instead the animal is galloping, we say that it has a phase of 0.75. And when it's bounding, we say it has a phase of 0 or 1. So you can see here that most of the steps when the animal is walking or trotting have a phase around 0.5, which means that the hind limbs or the forelimbs are alternating. So when we silence these neurons, what we end up seeing is a range of behaviors that involve non-alternation, even at walking speeds. So in the top animal, the forelimbs alternated, but the hind limbs kind of galloped. And in this middle animal, the forelimbs are hopping, the forelimbs are alternating, but the hind limbs are hopping. And in this third animal, even though it's at walking speed, it's actually just hopping. So the limbs, those steps would be represented up here. So you can see here that with the forelimbs and the hind limbs, when we turn these long propriospinal neurons off, we get a wide range of phase for the forelimbs and hind limbs, which means we've changed the interlimb coordination. Now, surprisingly, the animals still have a very stable walking. They do just fine. So what that tells to me what that tells me is that we're demonstrating that our hypothesis is correct. When we turn off neurons that carry information from the lumbar circuitry up to the forelimb circuitry, we can disrupt how both the forelimbs and the hind limbs behave during stepping. So this is spinal autonomy. This is something that we've known about for many years because of the work of many people, Serge Rossignol, Reggie Edgerton, showing that fully transected cats, when they put their hind limbs on the treadmill, they can train those hind limbs, retrain those hind limbs to step beautifully. That's spinal autonomy. So for spinal cord injury, all we have to do is get the CPG going again, right? That's what I have believed for many, many years. But we made a discovery in this study that was really quite remarkable. And that is that the disruption to stepping, I hope you can see my arrow, the disruption to stepping only occurred when the animals were walking on a flat, level surface that had good grip. When it was walking on a flat, level surface that was a little bit slippery, that disruption to stepping didn't occur. When the animal that was walking on a treadmill, it didn't occur. When the animal was walking A to B with its nose up, with a goal-directed stepping, we saw a disruption of the locomotion. When the animal put its nose down and started exploring, that disruption went away. So spinal autonomy, yes, but it's context specific. In this case, only when the animal is walking over a flat, level ground surface on something that had good grip, or for that matter, when the animal is swimming, which they don't use their forelimbs when they swim, it's a hind limb only. So this is really important, I think, because it shows that, yes, bottom-up spinal autonomy, the power of the central pattern generator is there, it's present, but it's context-specific, and it doesn't happen all the time. If the animal is exploring, if the animal is on something slippery, we don't see that kind of spinal autonomy. So the treadmill issue is a bit controversial because many labs, both human labs and preclinical labs that use mice and rats, they have the belief that the treadmill is a more spinal behavior. It's more automated, but that's not really supported by our work. So we argue that this supports a concept that I call supraspinal oversight. And it may mean that the supraspinal circuitry, the brain and the brainstem, is sending a patterned input saying right, left, to ensure that the stepping is being done in a safe fashion or during exploration because the animal is wanting to be able to rapidly move one way or the other. Or it could mean that the brain is simply sending a signal down to the spinal cord that's recruiting other circuitry to ensure a stable pattern, even though we've knocked out some of these neurons that project from the lumbar circuitry to the forelimb circuitry. So all of this suggests to me that the sensory input combined with goals influences circuitry function. Is the goal to get from A to B, and is the animal or person on a surface that has good grip and there's no obstacles? I think the spinal autonomy can be very high at that point. What about for somebody that's using a cane or walker, whether they need to or because they're hiking and they're using trekking poles? What about after injury and they're training in an exoskeleton? How do those things influence this beautiful dance that's going on between descending control and the spinal autonomy? When we train, what are we training for? Sometimes I see patients or subjects that are going through a training and they're looking down at their feet. They're thinking right, left, right, left. Sometimes they're looking ahead and they've got a goal to get to. volition is extremely important. Is the idea, let's get to the end of the corridor, or is the idea, let's get the right leg lifted, then let's get the left leg lifted? Does the training involve the pattern generating circuitry? Yes, I believe it does. And I think a lot of people, I hope a lot of you are thinking, what about transcutaneous stimulation? So let's talk about that. Transcutaneous stimulation or epidural stimulation, I think, accesses the pattern generating circuitry by activating, in part, all of these sensory fibers that bring the information, the sensory information, in a very region-specific fashion into the pattern generating circuitry from the muscles, from the joints, and from the skin. That allows the pattern generating circuitry, number one, excitation, and number two, information about how to generate the locomotor output. So what happens? Maybe the stimulation is tonic, like what the folks at Louisville use, Susie Arkema and team, and they're going to rely on the circuitry to turn that tonic stimulation into phasic output into the actual locomotor pattern by making full use of the pattern generating circuitry. But maybe the stimulation is patterned and is actually regionally specifically activating afferents to specifically activate specific muscle groups. So I'm going to demonstrate that here. Maybe the stimulation is being applied to specific sites that have access to the pattern generating circuitry and the flexor circuitry up here and the extensory circuitry down here. And I think that's what happens with the Cortine approach, where they use a computer to generate a pattern of input via epidural stimulation using smaller hotspots, and it requires closed loop control. However, maybe the circuitry, the stimulation is instead being driven by larger hotspots and using tonic input and driving the pattern generating circuitry to generate the actual pattern. There is less specificity here and more reliance on spinal circuitry, and it might be more amenable to a transcutaneous approach, like what was beautifully described by Chet Moritz a little bit earlier. So I'm challenging people to think about the bottom-up versus top-down control and how much they're involving the spinal cord circuitry and what they're doing. Because we're very short of time, I'm going to skip a few slides, and we're going to move on to some summary slides. So what I'd like to challenge people, both researchers, caregivers, and people with spinal cord injuries that are undergoing training, that want to get involved in a clinical trial or in some very specific training. What are we studying and what are we training for? Is treadmill the right way to do it, or should the training be done over ground? I can't answer that. All I'm suggesting is that for the rats, it seems like the spinally generated component of these things doesn't seem to be the same for treadmill versus over ground. What about the input, the surface that the person is walking on? What about the sensory input into the foot and into the limbs? Stimulation, how are we applying the modulation to the circuitry? Are we making use of the circuitry? Are we fighting against what the circuitry might want to do? Sometimes I wonder that some FES approaches where the muscle is being stimulated directly might not be working with the spinal circuitry in the most effective manner. And I didn't get to show you this data, but we have some data to suggest that the spinal circuitry after injury doesn't respond the same to training later after injury as it does soon after injury. So I try to encourage everybody to get started earlier, both in our training and also to study the differences between the circuitry early post-injury versus chronically. And then there are some really exciting things that are happening. Kareem Fouad's lab up in Edmonton and other labs, ways to reopen this window of opportunity for plasticity so that the spinal circuitry can actually respond to the input and generate and improve function over the long term. So these are the folks that are in the lab at the moment. And I want to emphasize the interdisciplinary program in translational neuroscience, our PhD program at the University of Louisville. And if any of you are thinking about doing a PhD, we would ask you to consider our program and to get in touch with you about it. So with that, I'm going to stop sharing, and thank you everybody for hanging around at the end of this marathon IOSCIRS, and I'm happy to answer any questions.
Thanks, David. And I like your hoarse voice, actually. I want to really quickly, and just kind of as an aside, I want to ask somewhere in the middle of your presentation, it had me wondering, and I'm guessing maybe some folks out there with SCI might be, when my son has a C5 injury, age A, and I built I put an I-beam in the garage, built a PT gym, and got a harness and walked him on a treadmill for a long time, 13, 16 months, something like that, in addition to a program that he was in where he walked on a treadmill 3 days a week for years.
Yeah.
Is he wasting his time?
I don't believe he's wasting his time at all, because even if the overground passing through space might be ideal for this specific context that we're talking about, the benefits of getting repetitive afferent input into the circuitry, of getting cyclical muscle activation, of energizing the system from the perspective of physiological exercise. All of those things are going to provide benefits that go way beyond the specific circuitry that we're talking about. We're talking about, I like like to say that our muscles are the biggest source of happy juice in our bodies. So if we can use our muscles, they're going to be releasing things that are helping everything. They're going to be helping our brain, our gut, our microbiome. Everything's going to benefit from that. So by no means is that wasting time. However, I will say that I think as we learn more and we can get more sophisticated about these things, we might be able to make small differences. Video that mimics passing through, going through space rather than being stationary. Ways of ensuring that there's arm swing. I think that we're quadrupeds that happen to walk upright. Ensuring that we're getting arm swing and coordination so that the afferent input going in is good. And I think, I'm just so excited about the potential of epidural and transcutaneous stimulation. I want to get on the horse and ride that horse as quickly as I can into the future, even though I don't actually study it myself. So I hope that helped.
Yeah, I was thinking about it, sort of this, in some ways in all three presentations, to me, there's a little theme of this like tension between trying to manufacture the environment and creatively exploiting the environment that exists, right?
Yep.
So,
yep.
And in some sense, like to what you said, I actually wanted to, and maybe Bethany, maybe it is, since we've gone long, it's time to maybe pull everybody in and address the questions. But I wanted to combine a few kind of in that theme when you talked about in combination. So for all of you, I guess one question, which I think a couple of people sort of lean towards, one is the use of transcutaneous stimulation in the acute stage of the injury, which would mean in a clinical setting. And even Caitlin, the conversation you were having with Gordon Minshall about the hypoxia-induced factor, using intermittent hypoxia to, coax those growth factors that you were talking about. And for those of you who don't know, intermittent hypoxia is sort of, again, creating the conditions to reduce intake of oxygen, to put stress on the body and then release, have an effect there. So in a broad sense, some of these questions about the combination of various strategies, I'm curious about for any of you. David, you just mentioned, get on the horse of applying some of these principles in rehab to transcutaneous stimulation, or I would expect epidural stimulation. Chet, the application of this, like, is this looking at clinic only and at certain stages of injury, or is this something people are going to take home? And then Caitlin, even in terms of cells, the combination of other interventions like electrical, spinal cord stimulation or optogenetics or other factors that might stimulate or promote growth and connectivity and function. So I throw it out to any of you. I hope I framed that in a way that sort of rolled up a few questions into one broad question.
So I'm going to say that if we could get on our inner spaceship, not an outer spaceship, but an inner spaceship, and fly into the spinal cord and see what's going on with the transcutaneous and epidural stimulation, we would want to then layer on top good cell therapies, good, proven drug therapies, in order to, prepare the circuitry and help the circuitry respond the best to this stimulation. I mean, that's our window into the circuitry, right? The afferents are the window into the circuitry. And clearly, the circuitry is sitting there waiting to respond. And I think, good cell therapies and good drug therapies, I mean, I'm excited to push forward with those as long as it's being done in a quality and responsible fashion. I'll be quiet now.
I'll layer on. I know this question was kind of pitched at Caitlin, but I do just want to mention, I totally agree with David. It's not just going to be epidural or transcutaneous stimulation. It's going to be the combination of cell therapies. There's going to be manipulations. We're currently testing optogenetics in our rats with spinal cord injury, seeing absolutely tremendous results, better than we've seen with electrical stimulation. So I don't think the problem is solved, and that's just the beginning, right? That optogenetics is stimulating existing cells. So what if Caitlin can put in stem cells, and then we can turn those on with optogenetics or some other factor? I think the sky's the limit.
Yeah, I think as a field for a long time, we've talked about combination strategies, and I think we're still identifying what those strategies are, when they need to be applied. And so we know a lot about individual strategies. We know some about combination strategies, and I think we're still trying to figure out what is that strategy and when do we use what strategy, right? The strategy for improving hand function where you lose the motor neurons might be very different than the strategy that you use at a thoracic level injury where the motor neurons control a small band of trunk musculature. And so those are some of the questions that we're still figuring out, but I think it's the stuff that David and Chet presented today is totally fascinating in terms of moving things forward. It's so cool.
So let me ask you a sort of deeper question, and hopefully you can, I hope it's not unfair of me to ask each of you that are kind of a little more in the earlier stage of discovery, right? And none of you represents the later stage. So again, it seems to be consensus from a lot of scientists that using a combinatorial approach is really going to be necessary to get maximal effect or benefit for people with spinal cord injury. But that pathway is very unclear, right? And we're also talking about strategies that are not yet available in the clinic, right? So can you comment on, is there anything that can be done now? And I would actually very specifically ask from each of you or any scientist who's listening, for us in the community as advocates and who are very motivated to accelerate innovations, what can we do about that? Is there something we can do as advocates to help start paving that pathway? Recognize that eventually we're going to have to combine therapies. So what can be done about that now?
All right, I'll start because it's a tricky question, Matthew. So I'll start, and then my colleagues can correct me. So I think what we need is an early win. We need something to get approved, even if it's not perfect. I'm kind of optimistic that something might be transcutaneous stimulation. We'll know in a year or two whether that trial was a success. Unfortunately, we just haven't had a home run yet. All of our stem cell trials have been pulled for economic reasons because the companies wanted to work on cancer instead. And that gets at what the advocates can do, I think. It's a small market for people with spinal cord injury, and that's a good thing, right? I don't want any more people with spinal cord injury. But what that means is that the advocates need to go out and raise both research funding and maybe some investment capital in these companies, and the Christopher and Dana Reeve Foundation has set up a nice model where donors through the Reeve Foundation are funding a lot of the onward trial, because at the end of the day, probably not going to make a lot of money treating people with spinal cord injury. So the standard venture capitalists aren't interested, but families of people with spinal cord injury certainly are. And that's helped to propel things to clinical trial when otherwise companies might just look the other way because it's not necessarily a profitable venture.
Yeah, I think that when we look at the field and the things that have moved forward to clinical trial, in many cases, they've been propelled by investigator will. So we can look at, Martin Schwab and Nogo, or Lisa McCarricker and Rowe, and Mary Bunge and Schwansel's, right? It's been the driving force of those investigators, which means that there's really big learning curves to get through the process, right? You can look at the COVID vaccine as an example of how hard it is to get something through clinical trials and into use, right? It's still not approved through the traditional mechanism, right? And that's been really, really rapid. And so there are challenges just even once the therapy is identified, getting it through that process and into persons with spinal cord injury. But I think, identifying what we think are those promising candidates and really pushing those through will be tremendously helpful. And Chet's right. We need some early wins before we can determine what the combination is going to be.
And I'm going to add, I'm extremely optimistic because the trajectory now is so much better even than 5 years ago. Basic preclinical researchers are now constantly talking about feasibility for translation, relevance to the people that are living with injuries and their caregivers. That's something that's in our heads all the time now. That has to be a good thing. And I think in Europe, they've set up basically a clinical trials incubator. And, we have some very, I think, important organizations in the US. Craig Nielsen is one of them that I also think can help perhaps set up a clinical trial incubator where we won't be come up against a roadblock because of the fact that you're not going to make a lot of money out at the end. And so I'm really optimistic.
Well, thanks. I think maybe since we're at 2 hours, we probably wrap it up here.
Yeah, well, I would like to say thank you so much to all of our speakers. Thank you to everyone who, I can see our viewer count. Thank you for hanging in there. It was a marathon, but I hope you found it useful. Again, speakers, thank you so much for giving your time and expertise. Thank you to everyone who's been participating in the chat. Before you leave, everybody, please, and I'll pop up our kind of parting slides. If you'll pull them up, Matthew, please.
Oh.
There is a survey that we have that we would love to hear from the people who have attended this session today, especially if you're not normally an IOSCIRS attendee. And also in the description, we have a ton of links to IOSCIRS, to NASCIC's engagement survey, to U2FP. We dropped in the chat, and I will drop it one more time. Maybe Chet, you can actually drop it again, the link for the study that you're enrolling for. So yes, thank you again to everybody. Thank you to Matthew. Thank you to our David, Chet, and Caitlin. And we, for the IOSCIRS, are going to be taking a little bit of a 1-month break, but we'll be back with some special programming in September with International Spinal Research Trust. And yes, maybe just I'll take all of our slides off, so I'll give everyone a chance to say goodbye to our lovely audience. Thank you to our speakers, and thank you to you, Matthew, and thank you to our audience.
Thanks, everybody.
Thank you.
Really appreciate it.
Take care.
Bye now.